Good morning, everyone. Happy Friday. I'm Senthil Sarvanamuthu, founder and CEO for ONS Bio LLC, and I welcome Dr. Shokufar and all the students and research colleagues from the In Situ Nanomedicine Lab within the Department of Bioengineering at the University of Illinois at Chicago. We thank Dr. Shokufar for providing us this opportunity to present today. This presentation is part of a series of outreach effort by ROKIT Healthcare, which is a South Korean-based biotech company focusing on regenerative medicine and genomics. And also, ROKIT is a manufacturer of 3D bioprinter organ regeneration platforms, INVIVO 4D and 4D6, and various other industrial 3D bioprinters. This webinar is brought to you today in partnership with Indicate Technologies, a Santa Clara-based distribution partner for ROKIT Healthcare, focusing on 3D bioprinting and measurement systems, and my company, ONS Bio, based out of Cleveland, Ohio. I'll be joined by Dae Lee from ROKIT Healthcare, Chris Kamimura from Indicate Technologies, and Parker Bovenberg, also from Indicate Technologies. Please keep your question and answers coming in through the Q&A chat window, and we will address the questions towards the end of the presentation. Today's presentation will be titled On-Demand Tissue Systems Engineering with 4D Bioprinting, and it will be delivered by Dae Lee from ROKIT Healthcare. Dae Lee is a senior bioconsultant and bioprinting specialist, and she's a former MIT researcher herself. Without further ado, over to Dae.
All right. Hey guys, good morning. It is my pleasure to be invited to speak today. Thank you to the UIC community for sort of hosting this webinar for us. And I would like to also acknowledge that ROKIT Healthcare is proud to partner with Indicate Technologies as well as ONS Bio in support of sort of evangelizing the technology of bioprinting across research and medicine. So again, my name is Dae Lee, and I'm a senior bioconsultant at ROKIT Healthcare. We're a company of organ regeneration platform technologies, especially specializing in 4D bioprinting and autologous therapy. Today, I'm going to be telling you about the benefits of bioprinting in the context of today's megatrends and events in science and healthcare. And in this talk, beyond the basics of bioprinting, I want to cover real-life applications and case studies of this technology around the world. And we'll actually be taking a look inside the research labs, the commercial industries, and even in the operating room where this technology has been used to actually treat patients. So Chris Kamimura and Parker from Indicate Technologies are here with us. Maybe before we begin, if you guys would like to say hello.
Hello, this is Senthil from ONS Bio.
Good morning, this is Chris Kamimura. We're here on the West Coast.
Good morning, this is Parker Bovenberg here on the West Coast.
Yep. And so Senthil, Chris, Parker, and I make a team of bioprinting and 3D printing specialists that you guys can always refer to for on-demand support in this front. All right, so without further ado, let's begin. So I want us to take a look around and see what's been happening in the news over the last 2 years. We for sure are living in a different world from just 5 years ago. These 5 events that I'm going to talk about really briefly will get us closer to the values for which bioprinting is rising to attention today. So in September 2018, we saw the feature news of a boy who had been living for 14 years with a new bladder that is made from his own cells. And these were seeded with a 3D inkjet machine into a decellularized pig bladder as a scaffold. And this was a work done by a famous enterprising tissue engineer and surgeon named Professor Anthony Atala, and many of you may know him already. In July 2019, the American Medical Association approved reimbursement codes for 3D printed surgical models and tools. While these are still temporary codes, which means the use of these codes needs to be tracked for 5 years at least until full official reimbursement, it's still progress that 3D printing is making into the hospital. And these are going to make the foundation for a clinical registry of patient 3D scan data and relevant 3D modelings, which will in turn serve as learning resources for an increasingly active implementation of 3D printing in biomedicine and possibly further on to 3D bioprinting of living materials. In September 2019, the US Environmental Protection Agency announced that it will eliminate all mammal testing by 2035, phasing out the use of animal experiments in cosmetic, drug development, and just basic science research. And so this is really quite a strong statement that's testifying to the fact that the research world is ready to make a transition to more humane research. And this really testifies to the urgency to find alternative solutions to animals for human research studies. In October 2019, ROKIT Healthcare and a diabetic foot ulcer specialty clinic in India put the bioprinter in the operating room to create a streamlined process of skin regeneration for diabetic foot ulcers, for which India is hit the hardest by. This new and alternative method for chronic wound healing promotes skin reconstruction using bioinks that are made from patients' own adipose tissue. These are printed into size-specific patches, applied directly onto wounds, and they integrate with the skin, basically priming for endogenous tissue regeneration. ROKIT Healthcare's mission here is to decrease or even eliminate amputation for patients with DFU, especially given the fact that the 5-year survival rate of DFU patients who have gone through amputation is actually lower than some cancer survivals. And last but not least, in the most recent months of 2020, COVID-19 really has changed everything about the way we live. Most notably, we've seen the pandemic disrupt the traditional centralized value chain in manufacturing. When hospitals got cut off from supplies of masks and ventilators because factories shut down, 3D printing came to the rescue to manufacture these supplies on demand and on site. So I've gone through 5 key issues or megatrends that we've seen in the news for the last 2 years. But what happens if we put this together? What's the take-home message from these global events? Well, I think that this means the technology for on-demand manufacturing, whether it is in industries, in research, or in medicine, is something that's going to rise to attention to meet the decentralized demands for manufacturing of products that we need. And it is for these connections to the real world that we are here today to talk about the 4D bioprinting technology. For these reasons, it may be worth paying attention to how the technology also further develops from this point on to impact our research and healthcare workflows today. Though bioprinting is getting a lot of attention in mainstream research recently, the origins of the technology actually go back 2 decades. It's not totally in its infancy as some may believe it to be. In fact, the number of publications around this technology have been growing exponentially since 2000. And today there's about 2,000 publications testifying to the benefits of the technology and evidence of the technology's usage across tissue engineering, biomaterial development, regenerative medicine, and more. And here, if you actually take a look at these tables, you see these blue hyperlinked numbers. These are all publications that are in the journals talking about the ideal choice and combination of cells and biomaterials for use for tissue-specific bioprinting applications. And so perhaps in the future, journal publications may require data from 3D human tissues like these in between 2D in vitro cultures and in vivo animal experiments. And we may imagine that future is not so far away. Then what exactly is 4D bioprinting that we're here about? 4D bioprinting is an additive manufacturing process in which cells, biopolymers, and growth factors are combined to create tissue-like structures that mimic native human tissues. Because these printed living tissues are often further incubated to have them proliferate, mature, and change over time, there is this dimension of time here. So this is why actually many researchers and scholars are calling it 4D bioprinting, not just 3D. The bioprinting technology consists of largely three components that you can see here on the screen: biomaterials, the bioprinter, and design. So first, let's take a look at the biomaterials. Unlike the traditional 3D printer, which uses hard plastics, metal, and synthetic materials to create non-living structures, a 4D bioprinter uses materials that are biocompatible, biodegradable, and derived both from natural and synthetic sources, and most often the applications are in healthcare. Naturally derived biomaterials include collagen, gelatin, elastin, chitin, which are some materials that come from the extracellular matrix of human or animal cells. Biomaterials can also come from plants or insects, like the examples of alginate, sericin, or cellulose, which come from seaweed, insects, and plants, respectively. And natural hydroxyapatite comes from mammalian bone. Synthetic biomaterials like PCl, PLA, PLGA, etc., are all biodegradable polymers that have been man-made, and they have enough strength and elasticity to mimic human tissues and often have references of use in already FDA-approved medical devices. And finally, you also, of course, have the composite biomaterials, which combine the benefits of naturally derived with synthetic in order to add certain mechanical or chemical properties to the materials as you want for your research purpose. And so in summary, these biomaterials can be soft, low viscosity hydrogels that you can combine with cells, or hard biodegradable plastics, which you would often use in, for example, bone tissue engineering. But when hydrogels are combined with cells for printing, these are called bioink. And bioinks basically serve as the ECM when you're trying to print cells outside the body, protecting them from shear stress of printing, providing them with nutrients and hydration, and giving the overall physiochemical support. So you can imagine that when selecting materials for bioink, there are several key criteria you want to consider. They can be broadly summarized into two broad categories. One is that the bioink should be able to extrude out of nozzle tips with enough gelatin kinetics, and they should be able to stack layer by layer structurally, whether they need some post-printing curing processes. Second, the bioink should be biocompatible. They should basically uphold high cell viability, and the cell toxicity would not be would be avoided in the bioinks. And so now, if we talk about the hardware, let me just mute this. There are various methods of bioprinting today and various hardwares, from inkjet-based, laser-assisted, and stereolithography. But we're gonna focus on extrusion today because it is the most common one used, over 70% of the bioprinting market today. And this is because cells printed from extrusion-based methods tend to have the higher cell viability compared to other methods. Now, one key component of a bioprinter hardware are the printheads that should be able to print not only hard, but soft materials. There are two ways that the printhead can print. One is scaffold laden, which means you basically make this hard matrix upon which cells can be printed over. And then the other one is scaffold free, meaning you don't print any hard matrix to serve as a house. You simply encapsulate cells inside a hydrogel and print it directly onto a platform. But a key component, of course, of a 4D bioprinter is the ability for the chamber to mimic a biosafety cabinet, because after all, you're dealing with living cells. So to optimize cell viability and functionality under the stress of printing, the chamber should have UV disinfection capabilities, the HEPA filter, as well as some type of bioreactor to provide optimal environments in terms of temperature, humidity, and CO2 levels for the mammalian cells. And finally, of the three components, we arrive at the software or the design components. And this means 3D models of the objects you want to print. 3D model files, which are known as by the global standard STL, these files can be obtained either by manual creation using software, design software, or they can be derived from CAD or MRI scans of physical human body parts. And these STL files then get processed with what's called a slicing software, which basically converts the original STL into standard machine language that describes how a 3D printer should print the job in a given three-dimensional space. And the information include XYZ positioning coordinates, printing speeds, infill patterns, etc. And you can see below images that demonstrate what an STL file is, the aqua blue object file on the left, and results of a G-code instruction with different infill densities from, for example, from 10% all the way to 100%. And having understood these components of a bioprinting platform, next we can understand the bioprinting process. The process principally involves these 5 key steps that I outlined here. But if I just summarize each of them briefly, preparation involves designing the experiment with a clear identification of the problem to address, and it includes selection of the biomaterials, cell types, and 3D models. In printing, you actually load the biomaterials onto some type of cartridge. You slice the STL files into G-codes and extrude the materials according to the digital model. And in post-printing, you basically would take the printed object and make some mechanical or chemical stimulation of it so as to create stable and functional structures for that material. Especially if you're dealing with hydrogel encapsulated cells, you often would need to cross-link the hydrogels through thermal, chemical, or light sources and make them more stable for further culturing. In evaluation, just as you would be familiar with in your own experiments already, the cultured printed tissues would be assessed for structural, morphological, or functional characterization in comparison with the living tissue standards. And that way you know that the 3D printed tissues are actually behaving as you may want, as you may expect. And finally, the application step, of course, involves making use of the printed products. And these could be for various purposes, such as disease modeling, in vitro testing methods, or in vivo tissue transplantation. And so if we take a look at an example of this same process in the context of cardiac patch engineering, it would look something like this. And this is taken directly from an actual publication, making use of bioprinting, extrusion-based bioprinting for cardiac patch printing. And I will let you take a look, a better look at these later on in your own free time. But the cool thing here is that when this bioprinted construct was transplanted onto a rat heart, they were able to then evaluate whether this printed patch works in sync with the heart by taking PCI-CT scans and doing a series of electrical conductivity and elastic modulus tests, which are interesting to overview directly here in this publication. But cardiac patch engineering is really just one of many, many downstream applications that is possible with bioprinting. And I want you to take home the message that bioprinting is really a platform technology. The cells, the biomaterial, the design that you have according to specific purposes can be realized. And it's been used in departments across tissue engineering, material development, drug discovery, veterinary medicine, toxicology, etc. But if I could summarize the bioprinting applications into 3 key areas, they would be the in vitro efficacy test model for toxicological testing. And this is especially important if we're trying to phase out animal experiments in the long run. Second is the in vitro study model for human tissue physiology and disease modeling. And finally is the in vivo model for transplantation and regeneration. And so the first of these 3 is the development of on-demand printed human tissues for testing the safety and efficacy of novel products, whether they may be drugs, cosmetics, or even food. And as you can imagine, an ideal study to evaluate the toxicity of any substance to humans requires an extremely long time and money in order to get the results for this substance's effects on human subjects, because you would have to go through in vitro animal studies as well as rounds of human subject studies. However, in order to represent all human subjects, this would be not only unrealistic, it's unethical. So the use of human relevant tissue models offer an alternative and a bridge between human clinical trials and animal studies with a standardized human tissue fabrication process and high throughput specific deposition as chemicals on these tissues. And here you see an image of bioprinted human skin equivalents for the use of cosmetic testing. Second is the in vitro study model of human tissue physiology and disease modeling. Cancer, for example, is a prime disease of such complexity and heterogeneity that bioprinting's ability to fabricate and visually map tissues in 3D would be extremely beneficial. This type of technology would get us to the most accurate understanding of the way that these tumor tissues develop and behave, and that would directly translate to higher accuracy in the development of cancer therapeutics. And finally, the in vivo model for transplantation and regeneration. One of the most common challenges in medicine, of course, is not having the sufficient number of tissues and organs to replace in the events of trauma and organ failures with age. Flat structures like skin and tubular structures like blood vessels and urethras may be relatively simple, but hollow non-tubular organs like bladders or pumping organs like hearts and lungs would be very difficult to replicate with standard manufacturing processes. And so for such highly complex organs, the bioprinting technology provides a kind of precision that surpasses human hands. And if these could be made with patients' own cells, the challenge of immune rejection and donor shortage with organ transplantation could be resolved once and for all. And here is a video of a bioprinted cardiac patch that is now beating without any external electrical input. so behaving as a cardiac muscle tissue should. And so in the context of all of these, ROKIT Healthcare and our community really are dedicated to providing the launchpads for organ regeneration, novel organ regeneration strategies and tissue engineering research projects that we seek to contribute to the advancement of breakthrough science in healthcare. And we've been dedicated to providing these offerings to over 25 countries around the world, and so far with over 250 user institutions of these systems. And we've been leading the introduction of bioprinters that have offered for the first time the world's first clean chamber bioprinters with HEPA and UV lamps. And we've also had the fortune to introduce bioprinters that are the world's first to completely integrate cell incubators and plasma sterilizers with 6-printed biofabrication technologies, which we'll overview as an online demo later on in the presentation. And so now let's get to the heart of the applications. Slides in the following are all summaries that I tried to put as abstracts of journal publications that have been published by the Dr. INVIVO bioprinter users around the world. And for more details on the journal publications that draw your attention, you can take a look at the link underneath each of the slides, and you can definitely take a better look at them. And so my goal here is to show you as diverse of a series of applications. So I won't go into very specific details, but if you guys have any questions about one specific application, please have your questions ready in the Q&A box and we'll answer them at the end. All right, so first we begin with the biomaterials research. And in fact, biomaterials, are the engines of tissue engineering, and the technology of bioprinting with this ability to basically print biomaterials in consistent manners in an automated way has really been playing a big role in characterization and development of novel materials. Actually, the journal that has the most number of publications with the keyword bioprinting is on the Journal of Biomaterials, which I found out just today. And in this particular study, the research group has developed a self-healing hydrogel with a composite of hyaluronate and chitosan. And basically, they create this consistent circular disc of the hydrogel. They cut them in half and are able to demonstrate here that these self-heal on their own at room temperature under 10 minutes. And what the research group shows that this self-healing biomaterial that is printed could not only be used as a patch for the delivery of drugs, but also it could be a bioengineered gel for the proliferation of cells like chondrocytes, as they test that this biomaterial is biocompatible with chondrocytes and promotes proliferation of stem cells into chondrocytes as well. Here we have an example of the use of hydrogel embossed platform for the mass production of stem cell spheroids. And with bioprinting, the group was able to basically print even layers of hydrogel that are seeded with adipose-derived stem cell spheroids, whose size could be controlled by the seeding density. And within the 3D printed construct, they demonstrate that the spheroids had excellent retention, anti-apoptotic, and pro-angiogenic properties. And these also had better osteogenic or chondrogenic differentiation than the 2D cultured stem cells. And accordingly, the group suggests that spheroid production using such bioprinted platforms could be used in fields such as cell therapy or ex vivo tissue reconstruction. In this study, the research group develops adipose tissues using PLCL, which is a application of PCL with basically a softer mechanical property closer to adipose tissue. And they have created size-specific patches that are combined with PLCL plus adipose-derived ECM for indications such as burns, trauma injuries, or post-mastectomy repairs. And they show in animal models that when implanted onto mice, this ECM and PLCL mixed customized patch was able to promote adipose tissue regeneration with macrophage infiltration as well as vascularization. And here is I think you can just take a look at this third figure in the center of the slide. Here, the research group combined hydrogel coating technology with 3D printed porous PCL scaffolds to demonstrate that you could culture and maintain salivary gland tissues. And the group says they look forward to applying this platform to create better human models for other branching epithelial organs as well. Now we've talked about biomaterials and some examples of soft tissues. Bioprinting has also demonstrated efficacy in creating beading models as well, most notably heart tissues and muscle tissues, musculoskeletal tissues. So this one is about creating in vitro models of pathological cardiac tissues using bioartificial scaffolds. And the key thing here is that this group from Italy used different patterns of PCL scaffolds, some strands in different angles, to create tissues that mimic different electromechanical properties and therefore different stages in the progression of ischemic heart disease. In this case study, cell suspensions of musculoskeletal cells of mice were printed around 2 PCL posts, also of customized size and shapes, to create muscle models for drug testing. And here, iPSC-derived cardiac spheroids were combined with endothelial cells, fibroblasts, and muscle cells into 3D printable spheroids. And these spheroids are then used as biomaterials for printing onto a hydrogel patch to ultimately create a vascularized model of the cardiac tissue, which is a step forward from the previous cardiac tissue version that I showed. And of course, in disease modelings, bioprinting has been used to advance more physiologically relevant human tissue models. In this study by Jungwon University of Korea, as well as Wake Forest Regenerative Medicine Center in the US, bioprinting was used to develop 3D human bladder cancer models and compare them with 2D models. And basically, in evaluations of those two models' behavior to 2 types of chemotherapies called rapamycin and BCG, the group found that 3D tumor models basically demonstrated a much more aggressive behavior of proliferation, and they had greater resistance to these drugs and expressed them in different profile expression. So the group concludes that using 3D models of cancer is crucial to really better assess a drug's efficacy to cancer. And here we go a little step further than before to combine several cell types into a tumor model. First, we have a bioprinted layer of lung tissues, lung fibroblasts, with HUVEC representing blood vessels in a composite bioink of alginate, fibrinogen, and gelatin. And you can see that these different biomaterials are combined to achieve a certain biochemical and mechanical characteristic. Then multicellular tumor spheroids of glioblastoma cells were seeded onto this vascularized lung tissue, and they were incubated, resulting in vascularized tumor models also for physiology studies and drug screening studies. Bioprinting has also opened doors to a novel QR-coded drug system. And if you can take a look at the figure A and B, you can see how thin that is, a thin oral dispersible film that is to replace some of the drugs that are hard to swallow. But not only that, this type of 3D printed drug has the potential to support patient compliance because it's not only a drug that is easy to swallow, but it is also highly informative because it's a QR code itself. And the ability to deliver information about the patient's prescription right upon scanning the drug could both reduce misconsumption and misadministration of medicine. And the fact that there is a trend these days of pharma companies replacing anti-cancer injections with less invasive oral dispersible films, we can see that bioprinting has applications to seek a more effective and minimally invasive therapy as well. And here, I will just briefly overview that bioprinting has been used to do microbial studies as well. They basically created an egg-shaped bioresponsive device where the research group was able to specifically deposit microbial organisms into specific desired positions in desired locations. And they were able to include carbon nanofiber led materials into this device so that activities by these microbes could be monitored. And the application for such a device could be in wastewater, for the studies of wastewater treatment, or the basic microbial biosynthesis or metabolism studies. And interestingly, bioprinting has found application not just in human tissue and material research, but it has also found applications in customized fabrication of devices. Left here right now is the creation of an ergonomic bracelet using petrochemical TPE combined with nanocellulose so that it serves as a thermal therapeutic device that can heat as well as cool. And here we have an example of the development of radioisotope adsorbents with better porosity control and cheaper customized production compared to the regular manufacturing process being used in nuclear adsorbent production. But perhaps the most noteworthy applications are those that take this technology off the bench and into the clinic. As we've seen before, ROKIT Healthcare has been leading a project to reduce amputations in diabetic foot ulcer patients by offering a completely regenerative solution. And this is one that leverages the patient's own autologous solutions with the size-specific bioprinting technology. And the result is an in-clinic bedside solution. And based on ROKIT Healthcare's clinical studies, results in India and Korea, it took on average 4 weeks to close the wounds that had otherwise stayed open for months to years. And these immense findings have continued on to more global clinical studies that are in line currently in the US, Turkey, and Italy in the upcoming year. And we envision that this therapy is not only restricted to diabetic foot ulcers, of course, it can be applied to more chronic wound indications, whether from burns or scars to bed sores. And one can imagine that such a therapy can be called an integrated 3D scan print therapy, a novel one of its kind that I believe would become more realistic in the near future. This takes the patient image and translates it into a personalized regenerative solution right by the bedside. And here in this video, I wanted to show you what that entire process looks like in a few seconds. So don't be surprised, this is a model, a silicone model of the foot with wounds that are represented by these incisions in the silicone model. But the idea is that a 3D scanner scans the wound sizes and is able to directly convert these images into STL files using this automated software. Let me just fast forward a bit. And this file that is created with specific wound measurements and specific triggers for scaffold printing would trigger the bioprinter to print a scaffold with FDA-approved biopolymers, and then it deposits patients' autologous solutions into the scaffold, creating a patch that can be directly applied to the wound. And this is actually an image from our India clinical study in India with DFU patients. And so DFU skin regeneration is not the only application. There are other applications for clinical product development currently in the pipeline with ROKIT Healthcare community. Here is an example of customized bone implant manufacturing. And this process has already received approval for distribution by the Korean equivalent of the FDA, which is called the MFDS. And here, in joint research with Mass General Hospital, ROKIT Healthcare is conducting studies for cartilage regeneration. As you know, cartilage stem cell-based injections have the disadvantage of having some of those stem cells disperse after they have been injected into the body. But having a scaffold-laden stem cell-based matrix that is inserted into the defect area has the advantage of localizing the effects of those stem cells into the defect. And so far, with 3 rounds in Beagle experiments, we have seen localized hyaline cartilage regeneration, and we have observed beagles recovering walking functions. And so in the last few minutes, I would like to take you through a demonstration of ROKIT Healthcare's bioprinters, just wanting you to see with your own eyes, even though we do not have an offline demonstration right now, at least with videos and images, what bioprinters actually look like. So here is our first generation Dr. INVIVO 4D that has the capacity of printheads to print with hydrogels and bioinks with cells. It's got the capacity to print with filament biopolymers with a filament extruder. And it also has the ability to accommodate pellets, powders, and pastes in a hot melt pneumatic dispenser. And the key thing here is that because biomaterials, one of the key primers of biomaterial actions is temperature, all of these printheads together are able to cover from -4 degrees to 350 degrees to open up opportunities for various biomaterials to be used. And of course, as the world's first clean chamber bioprinter, it's got its own closed chamber with the door, with UV lights and HEPA filters to keep a clean setting inside. But something we have learned over the years, especially with the clinical studies happening around the world, we've listened to some advice from doctors themselves who have said that in order for bioprinting to really translate to the operating room, it needs to meet certain requirements for organ regeneration. And that is, number one, we would benefit from being able to directly apply biomaterials on curved surfaces of the body. In other words, in situ printing technology of some sort. Second is disinfection is not enough. Often we need sterilization so that the device itself is not only sterile, it should be able to sterilize implantable devices that it creates inside the chamber. But foremost, the idea that what we are printing right now with 4D bioprinters are not non-living stuff. These are living cells and tissues that require certain conditions of their native environment, such as pH, CO2 tensions, humidity, and temperature that are close to the native conditions of mammalian cells. So if we could combine all of these three requirements into one, that would be the ideal organ regenerator for the future of research and medicine. And so keeping to the best standards of what we can within these three requirements, we have now created and launched Dr. INVIVO 46. And it combines the 6 printed technologies that are able to accommodate various materials from hydrogels to hard plastics, taking the benefits of the 42 technology. And it's got a built-in incubator and sterilizer so that we optimize cell viability even after the cells have been printed. The high throughput mechanism of printing onto well plates has been added to the bioprinter so that the development of toxicity models or drug delivery devices in well plates is possible for many biological replicates in one experiment. And of course, the diverse material coverage is maintained here in Dr. INVIVO 46, just as in Dr. INVIVO 42. But the idea that some of these systems have already been used in the operating room and in GMP facilities, we can offer a fast track to medical commercialization, and that is positive news for rather state-of-the-art technologies like bioprinting in today's world. And finally, here is a sort of a video demonstration of the user interface of the software that you use to basically control the printer. And so the printer can not only be controlled from PC, but it can be controlled on mobile devices like phone and this tablet. And so in summary, what is the essence of bioprinting? If you're gonna take home one message, it is this. 3 essential key points of bioprinting are: number 1, we've got automation. Whatever we've been doing manually with hands, we can now replace automation provided by 4D bioprinters. Number 2, as much as we understand that our human cells and tissues exist in 3 dimensions, we now have a technology that can, at least at its most optimal level, replicate those structures in 3 dimensions. And that's going to directly impact the physiochemical properties of these tissues that you create. And finally, is the idea of on-demand fabrication. For a long time, we have depended on models or animal experiments that are either too expensive or not ideal for the particular study that we pursue. But if we have bioprinting capabilities in each one of our labs, the potential to create your own samples in a standardized fashion would open up a lot of doors in research avenues. And so imagine that bioprinting facilities could be set up in every region of the world, and the technology has the potential to completely transform regional biomanufacturing value chains, just as COVID-19 gave us a hint of its needs. And in fact, it's not surprising that bioprinting centers of excellence have already been arising, both at the national and international levels over the last several years already. So with all that we've shared so far, we can see that bioprinting is not only a revolution that's underway for the way we create and apply model systems for answering basic biological questions, is also driving a revolution in the way we practice medicine and make the most benefit out of technologies in healthcare therapies. Instead of using systems that only seek to treat symptoms using small molecules or monotherapies, the technology of bioprinting, especially with the potential to be by the bedside, really shifts focus toward harnessing the body's own healing potential inherent in the stem cells and immune cells inside our body. And this is the very vision and core mission with which ROKIT Healthcare works with our community. And so thank you so much for your participation in today's webinar. And I am, in fact, quite encouraged to see that the participants in this webinar can potentially share with us in this vision to revolutionize research and medicine with the technology that we can palpably use right from now. So thank you so much. And I will now open up the floor for any questions.
Thank you, Dai, for that informative and insightful presentation. Do we have any questions? I don't see any in the Q&A box.
So I actually see one in the Q&A box, but it has already been answered. Yeah,
Yes.
is there research being done in tissue regeneration for diseased plants? Would the biomaterials needed for plant cells be different than the ones listed in this presentation? And Santil from ONS Bio has answered that, yeah, plant industries, plant research, there aren't as many users as in veterinary medicine or human regenerative medicine, particularly because I've talked to some plant biologists in the past few months, and there isn't as much of a lack of samples from plants. It's not as hard to get biological samples from plants around us to use for research as it is from animals or humans. So it seems like bioprinting has a bit more room to go before it applies to plant tissue engineering. But one application we can find is that some rare plants that exist in extreme environmental conditions where it's hard for humans to access, if we can take cellular biomaterials from those plants and replicate them in the lab using bioprinting, that might open doors for us to have better access to study rare plants in extreme conditions.
Dai, there is another question from Dr. Shokufar. Again, I'll use this opportunity to thank her for giving us this opportunity and arranging this today. Thank you. The question is, how do you ensure the vascularization is adequately happening in thicker samples? And I think that's a very good question.
Yeah. Whether vascularization or any other sort of functional activities that we want to see in these printed tissues, researchers often make use of histology, or what do you call those? Histology, as well as immunofluorescence microscopy to basically analyze these thicker samples, just as you would do in regular experiments. But better than that, in thicker samples, you would make use of 3D scanning and 3D imaging capabilities in order to ensure that these types of processes are adequately happening. But in vivo, you would assess the process of vascularization by the way that they integrate and show show certain protein expressions after they have been harvested from the animals to see if that vascularization actually has adequately happened and taken place over the several days that you have had those tissues implanted into an animal body.
Sure. I hope that answered that question. There is one other question here. Is there any limitations to 4D bioprinting from Tamara? Would you like to go first, Dai?
Yeah, sure. Certainly there are limitations to the bioprinting technology. And these are several challenges that still move the technology forward to progress and mature. One limitation is that most 4D bioprinting technology platforms that are available today are limited to microstructure development, mainly because of the print bed size that is available. So if you actually, if you want to translate the technology to more full voluminous organ systems that are the size to fit our human body, the ability to manufacture these systems in large dimensions would require much more room for research and development. And so because of this, there's a lot of the bioprinting centers of excellence are focusing on creation of bioreactor systems that can well harvest these full voluminous bioprinted organ systems as well. Another limitation to the bioprinting technology is, of course, just finding the ideal bio ink that has the perfect biocompatibility, the perfect printability, and is able to implant into the human body with FDA approval. To be honest, there aren't very many biomaterials that are FDA approved for use in clinical application. So a lot of the research focuses on developing more of these biomaterials that can work as the engines for the 4D bioprinting technology.
And if I may add to that, a question from Tamara, which you just answered. Vascularization, going back to the question from Dr. Shokufar, that could also be a limitation to any type of bioprinting, be it 4D or 5D or 6D, because there could be, it's conceivable that there will be a limit to how fine a resolution we can get down to printing, which may not be optimal for the finest of capillaries that you find in tissues and organs. So that could be a challenge unless we figure out a way to find materials and techniques to recapitulate those fine capillary systems when we try to print functional organs and tissues. So there is a follow-on question from Dr. Shokufar about the previous question on vascularization. How would you design the system to make sure it's functional? Dae?
Can you repeat the question?
Yeah, the question is, how would you design the system to make sure it is functional, meaning the tissue is functional?
Well, there are several ways that you can test that your printed tissue is functional. But at the very basic level, if we go through the process that bioprinting researchers would take, you would take your tissue, or you will take your bio ink that has the hydrogel with your cells and print them out, making sure that the cells exhibit high cell viability in their hydrogel environment. So the initial thing is, of course, the cell viability test. And then for certain stem cells in those hydrogels, you would also do tests for making sure that those stem cells differentiate into the cell types of your choice adequately within that hydrogel system. And then once you have printed tissues in your experiment, those printed tissues can be further analyzed using various microscopy techniques, like the ones I mentioned before with fluorescent labeling of certain protein markers. For example, they can also be implanted in vivo into animal models to make sure that if these printed tissues are supposed to serve as primers for endogenous tissue regeneration, whether they actually make that happen in vivo in the animal models. Yeah, and like I think as we addressed a few questions ago, some of these thicker implanted tissues get analyzed using confocal microscopy as well for these 3D printed tissues.
Okay, thank you. Maybe in the interest of time, we'll take the last question here from Mila. Thank you for your talk today. It's great to see the advances in the field. On a fundamental level, how does your system address the challenges of oxygen and nutrient delivery to the cells in a 3D model?
Yeah. Essentially, there are two ways that bioprinting allows us to address the challenges of oxygen nutrient delivery. First is by the development of porous structures. So if you can imagine 3D spheroids, one of the challenges there is the development of necrotic cores, because you basically have these cell aggregates that combine into a sphere-like structure with no channels that allow delivery of the media nutrients into the very core of those spheroids. The way that bioprinting allows us to overcome the challenge is by making a scaffold that has pores of certain sizes that you want. And of course, those pore sizes would differ based on the tissues that you use. But having that scaffold at the beginning allows you to create a structure that already has channels and holes to which media nutrients can flow into. And then upon these scaffolds, cells stick to and develop tissues that then still retain the porous structures. And the second way that bioprinting addresses this challenge is by basically having nutrients inside the bio ink that is supposed to encapsulate those cells when you're printing them. And the bioprinter chamber can also provide its own environment optimal to mammalian cell culture, such as, although not directly related to oxygen nutrient delivery, but it can help with the overall cell growth and maturation by providing a chamber full of temperature, 37-degree temperature control or CO2 level control and humidity control, so that overall the cell viability is upheld.
Okay. All right. I think with that, we can conclude in the interest of time. While we can be here for another half an hour or more, but I guess the most other attendees may not be able to do so. All right. Thank you very much, everyone who participated today and made the session lively. Again, I thank Dr. Shokofer for giving us this opportunity and making this webinar possible. And If you have any further questions, please feel free to email me directly or Chris or Dave, and we'll be happy to answer them offline.